In this lesson, you will learn how gene expression is regulated in prokaryotic cells. The process of gene regulation is important for cellular activity. All genes in a cell are not turned on at the same time. If you think about it, we have like 30,000 genes in the human genome. If they were all turned on at the same time in all of our cells, chaos would ensue. We would never be able to have different cell types because cell types are regulated by which genes are turned on or turned off. So in this lesson, you will learn how do organisms control which genes are on and which genes are off in a cell. The first way to control gene regulation is by transcriptional control. Controlling the production of mRNA of a particular gene is going to determine whether or not a protein is produced. If you don't make mRNA for a gene, then you can never get the protein that would be created from that gene. Therefore, if you don't produce the mRNA, the gene is turned off. If the cell needs to get the gene expressed, if the cell needs the protein of that gene, then we can turn on transcription, make the mRNA, and then the mRNA will go to a ribosome to create a protein. In prokaryotes, because they don't have a nucleus, transcription and translation are both going to take place in the cytoplasm of the cell. Because of that, the genes are actually arranged in what are called operons in a prokaryotic cell, and the operon is a set of structural genes lined up together all in the genome, and they can all be turned on or turned off at the same time. An example of an operon in prokaryotes is the lac operon. The lac operon is turned on when lactose needs to be broken down as a sugar source. If glucose is no longer available to the bacterial cell for cellular respiration, then lactose can be used. Lactose is made up of glucose and galactose, so if we break down lactose, we can get the glucose out of it. So in high galactose concentrations, then the operon is turned on. The genes are transcribed to make mRNA that will then in turn make proteins and enzymes that are responsible for breaking lactose down. But if there's no lactose in the cell, then the operon does not need to be on. It's actually turned off so that those enzymes and those proteins that are responsible for lactose breakdown aren't just chilling in the cell, wasting space and wasting energy. So again, operons are a unit of linked genes that are with related functions, and they're expressed together. Operons contain what are called structural genes. Structural genes are genes for a metabolic pathway that are grouped on a chromosome and then transcribed at the same time. These genes are linked together on the chromosome because they all have the same function or the same related function. So they can be turned on together all at the same time and turned off together all at the same time. This is very beneficial for a prokaryote since they have all of their genome in one single circular chromosome, so it makes things very nice and compact for them. Like I said before, the lac operon is an example of transcriptional control of gene regulation. Again, this is in prokaryotes only. Lac operons and operons in general are not in eukaryotes, only in prokaryotes. And they actually discovered operons in E. coli. Okay, this is how the lac operon works. Again, when lactose is present in the cell, then the operon is turned on. What happens, and you'll notice here, is that there is a protein in red. See the red protein there? That is an inducer molecule. Or excuse me, the protein is not an inducer molecule. The protein is a repressor. Normally, that protein could come up and it could bind this area right here in the genome, and it could stop RNA polymerase from progressing along the genome and transcribing these genes, LAC-Z, LAC-Y, and LAC-A. But what happens is when lactose is present, this is called an inducer molecule, because it binds to the repressor, which is that red thing again, that's the repressor that represses the expression of the lactose genes. The inducer, lactose, can bind this repressor, making it change shape. So this inactive repressor can no longer bind to this region right here. It cannot bind. And when it doesn't bind, then RNA polymerase is able to progress along the genome, transcribing those genes, LAC-Z, LAC-Y, and LAC-A. And that will produce the products beta-galactosidase, permease, and transacetylase. And these are three enzymes that are going to be responsible for breaking down lactose. So again, the lac operon is an inducible operon. It can be turned on when a molecule called an inducer, in this case it's lactose, binds to a repressor and the repressor falls off the genome. When the repressor falls off the genome, RNA polymerase can progress and it can transcribe these genes into the protein products that are necessary for lactose breakdown. So there's different parts to an operon. One of the parts of the operon is the regulator gene. The regulator gene is not usually part of the operon itself, so it's not usually in sequence with the other structural genes. But the regulator gene is going to code for a protein that acts as a repressor. In our case, the regulator gene would have been the gene that's responsible for creating that red repressor that you saw in the lac operon example. The repressor controls whether the operon is active or not. So when the repressor is bound to the DNA, it's going to stop transcription of those genes. And when the repressor is bound to the DNA, the operon is off. As soon as the repressor goes away from the DNA, then the operon can turn on because RNA polymerase can now progress down the genome, transcribing mRNA into proteins. The promoter sequence is where RNA polymerase begins transcription. You guys already know about promoter sequences. You saw them with the TATA box when we talked about transcription. The operator is the segment of DNA where the repressor protein binds. So that little yellow area that you saw back with the lac operon where the red repressor molecule could bind, that's called the operator. So when there's no repressor bound to the operator, then the operon is on. RNA polymerase can attach to the promoter and it can transcribe the genes that are in the operon. But if the repressor is bound to the operator, then RNA polymerase cannot bind to the promoter and it cannot transcribe the genes. So like I said before, the lac operon is an inducible operon. It's always off in the cell until it needs to be turned on. It'll be turned on when the lactose acts as an inducer molecule, binding to the repressor, changing its shape, which makes it fall off the DNA, and then allows for RNA polymerase to bind to the DNA and transcribe the genes. We call this an inducible operon because to induce something means to turn it on, to make it happen. Another example that we'll learn about later is called the trip operon. The trip operon is a repressible operon. I should say a repressible operon, not enzyme. A repressible operon is an operon that's always going to be turned on unless something turns it off. So a repressible operon is going to be turned off when a co-repressor molecule binds to the DNA and turns the operon off. It blocks the transcription of the genes that are part of the operon, and then that operon can be turned off. We'll talk more about trip operon later. On Friday, we will do a simulation looking at the lac operon on the laptop computers in the library, but we will meet in class first. We'll discuss a couple things, and then we'll head down to the library. We'll also do an activity with repressible operons as well. Okay, have a good day.